Our protocol investigates fungal infection from both the host and pathogen perspectives to identify interactions between the biological systems that are critical for disease. We can detect both fungal and host proteins in a single experiment and identify fungal proteins only produced during infection, representing novel infection-associated proteins.
Although we focus on treating fungal infections by uncovering novel antivirulent strategies for treatment, our proteomics and bioinformatics pipeline is universal and could be applied to many biological systems. Our approach lays the foundation for studies of different pathogens and numerous immune responses and can be used to provide new insights into the relationship between Cryptococcus and the innate immune system.
The macrophage response to pathogens and detecting enough fungal proteins are important. Therefore, testing and optimizing MOIs and time points for each species are recommended. As macrophage and fungal cell culture can be challenging, knowing what to expect after co-culture is important. Visualization provides the researcher with confidence in implementing the procedure.
To prepare fungal cells for a macrophage infection, collect and centrifuge C. neoformans cells from a mid-long phase culture, followed by 3 gentle washes with 1 milliliter of room temperature PBS under the same centrifuge conditions. After the last wash, resuspend the fungal cells at a 1.2 times 10 to the 8th cells per milliliter of antibiotic-free cell culture medium concentration, and add 1 milliliter of fungal cell suspension to each of 4 wells of a 70 to 80% confluent 6-well macrophage culture plate.